Yeah, am I audible? Am I audible? Can we start?
Yes, sir.
Okay. All of you can see my screen. Okay, so good morning. We will talk about a brief discussion on general microbiology today. General microbiology, which includes the presentation about, in general, introduction to microbiology, bacterial cultures, culture medias, sterilization, and disinfection. Okay. Now, the whole credit for the observation and the description of the bacteria goes to Anthony van Leeuwenhoek. This is very known to all of you. And Robert Hooke has confirmed his observations. Louis Pasteur is considered as the father of microbiology and life attenuated vaccines. Joseph Lister, father of antiseptic surgery, was a person who has introduced Listerine, if you all remember. Robert Koch, considered as father of bacteriology, and I think you all are very much aware of Koch postulates. Then Paul Ehrlich is considered as father of chemotherapy. So these are the people who are responsible: Anton van Leeuwenhoek, Robert Hooke, Louis Pasteur, Joseph Lister, Robert Koch, and Paul Ehrlich. Now, in general, whenever we start with microbiology, it is mandatory for you all to understand the basic distinctive features between a prokaryote and a eukaryotic cell. You all are very much aware that prokaryotic cells, this nucleus is absent. The membrane-bound organelles is absent. The ribosomes is 70S, whereas eukaryote it is 80S. The cell wall has more of peptidoglycan. Eukaryotes have no peptidoglycan. Then the mitotic division is there more in eukaryotic cells. Histones protein associated with the DNA of the nucleus is present in the eukaryotic cell. Number of chromosomes is more than one in eukaryotic cell. Cell membrane composition is no sterols in prokaryote. Sterols are present in eukaryotic cell. Usually prokaryotic are unicellular, like you can have an example of amoeba, plasmodium, and all those things. And the size of the cell is very small. It is 1 to 5 micrometer. So you may get a point like which among the following are the features of prokaryotic cell. So something which you should be very familiar with is the ribosome is 70S. The peptidoglycan is present in prokaryote, single chromosome, single cell, and no sterols in the cell membrane composition except in mycoplasma. This is very, very important for you to understand the basic difference between the prokaryotic cell and a eukaryotic cell. Now, when you study the bacteria, you have got various methods in which you can study the bacteria. You can study the bacteria in an unstained preparation, which is called as a wet preparation. Example, a hanging drop preparation for a bacterial mobility. means the bacteria which are mobile, example Treponema, you can observe them in a wet preparation or a hanging drop preparation. This is an important question. You even use dark field microscopy for evaluating spirochetes. For a stained preparation, like a simple stain, you have got methylene blue. For a negative staining, you have got bacterial capsules. Means when you're doing negative staining, or for what do you do negative staining? It is the bacterial capsules. Impregnation methods are used for demonstrating flagella, and differential stains is the best example I can give you is an acid fast stain, which is basically used to observe Mycobacteria tuberculosis. So you have got a simple stain, means a single stain, negative stains, impregnation methods, and differential stains, which are used, dark field microscopy, or wet preparations like hanging drop preparations, which are used for determining or observing the bacterial mobility. These are the various structures of the bacteria. They may be present in the form of a cocci. The cocci can be in the form of bunch of grapes, example Staphylococcus aureus. They can be present in a set of pairs like Streptococcus pneumoniae, you can call them as pneumococcus. Chains, Streptococcus pyogenes, right? Bacilli or rods, you can see Bacillus anthracis, or you can see Haemophilus influenza. Spiral curved, comma shaped Vibrio cholera, or you can see something like a spiral, like a worm, is like name Borrelia. This could be gram positive or gram negative. The one in purple is gram positive. The one in the pinkish color is gram negative. There. So this is how you classify the organisms based upon simple staining, gram positive or gram negative, or morphological classification, which can be in the form of a cocci, bunch of grapes, chain, and this is very important. What is the taxonomy of a bacteria? A taxonomy of a bacteria means the domain being the bacteria. Then you have a phylum. You should know the hierarchy of the presence. So from domain to phylum to class, order. You can write. You can be very clear that whenever you write about order, it ends with ales, like Enterobacteriales, ALES. When it's a family, it is AC, Enterobacteriaceae. Genus, you know very well, is Escherichia, and species is coli. In species, you have got various strains. Sometimes you use the word strain, sometimes you can use the word subtype, serotype. You have a particular number. Like for your coronavirus, also you have got alpha, beta, gamma. These are all subtypes. Inside, you have got again subtypes. So they are subtypes or strains or subspecies. So it starts from domain, phylum, class, order, family, genus, and species. This is the cell structure of a bacteria. You can consider it to be a bacteria, means a prokaryote. You have got a spore, you have got a DNA, you have got a ribosome 70S, you have got a flagellum, you have got small granules. Can you see something is missing in this? When I say it as a prokaryote, you can see there is something which is missing. That is the nucleus. But you will say, but you have to observe that there is DNA. But the difference between the bacterial DNA and the eukaryotic DNA is the DNA is free, single. That is, they are not bonded to form chromatin. Okay, and there is no histone. If you see the structure of a prokaryotic cell, it can be divided into gram stable, if it is able to retain the original primary color, which is crystal violet, it is called as gram-positive. Bacteria which will not retain the primary stain and will take over the next stain, they are gram-negative. Gram stain attaches to the peptidoglycan in a bacterial cell wall. In a gram-negative bacteria and a gram-positive bacteria, the difference is where the peptidoglycan is there. Because the gram stain attaches to the peptidoglycan. In gram-negative bacteria, peptidoglycan is protected by an outer membrane. That is why it is not able to retain the primary color. This is the difference between gram-positive and gram-negative. Gram-positive, there is no outer membrane. There is a periplasmic space definitely present. There is peptidoglycan, teichoic acid, lipoteichoic acid. Peptidoglycan is there. Permeability of molecules is high. Okay, whereas in gram-negative, the number of layers are two. You have an inner and outer. You have a lipopolysaccharide layer. You have a peptidoglycan layer. The periplasmic space is less. The peptidoglycan is also less, and the penetrability is also less. Please see the difference and try to understand. You have a cell membrane. You know what a cell membrane is made up of. What is cell membrane made up of? Every cell membrane is made up of lipid and protein. It's the lipoproteinaceous layer. Outside, you can see a cell wall. When you try to understand this diagram. This is one of the most important aspect of microbiology to understand the total microbiology, not only general microbiology, but also periodontal microbiology. Actually, I should say more for periodontal microbiology. You have a cell membrane, you have a cell wall. The cell wall is made up of peptidoglycan, as I mentioned you before. Peptidoglycan, then you can see teichoic acid and lipoteichoic acid. You will see that the periplasmic space is also less, but peptidoglycan layer is very, very thick. In gram-negative, you see similarly one more wall, outer membrane also. Between you see less thickened peptidoglycans and you see periplasmic space. So you have got a bilayer phospholipid layer. In between you have got peptidoglycan. That is why the penetrability is less. You have got proteins and you can see something called as an O antigen. These are all part of the gram-negative bacteria. Your O antigen is all what is going to form an endotoxin because gram-positive here. Gram-positive, you have got plasma membrane and you have got peptidoglycan. Peptidoglycan is thick. Gram-negative, you have got outer membrane, inner membrane, peptidoglycan in between, and you have got a periplasmic space. You have got a periplasmic space which is present in a gram-negative cell wall. Okay. They are bacteria with defective cell wall. Like mycoplasma doesn't have cell wall. That is very important for you to understand. Mycoplasma do not have cell wall. This is a very, very important question which is asked in exam. Like which among the form of bacteria have no cell wall? Answer is mycoplasma. Then you have got protoplast. Protoplast is the gram-positive bacteria with a defective cell wall, which is killed by an antibiotic. That means whenever the penicillins are given, what will penicillin do? Penicillin mechanism is inhibiting the cell wall synthesis. So when it inhibits the cell wall synthesis, the cell wall appears little abnormal, defective, but not complete. Such a cell wall of a gram-positive bacteria is called as protoplast. P for penicillins, P for protoplast. Spiroplast is the gram negative, which will have an effect from an antibiotic. It can be again penicillin, and it's called as spiroplast. It may L forms can be gram positive or negative bacteria lose their cell wall due to mutation. They're able to grow and divide or multiply, and this is possible in a different type of media. And this is how in a medium they shrink, and that form of the bacteria is called as L form of bacteria. So they lose the cell wall due to mutation. It can happen either in a hypertonic or hypotonic or isotonic anytime in order to retain its contents and to not release them. Okay, so in this medium, shrinking may be happening. L form bacteria are also known as L phase bacteria or L phase variants or a cell wall deficient bacteria are strains of bacteria that lack cell wall. It was first isolated by a Nobel Prize winner, and they were named, and was after the Lister Institute in London, where she was working. Two types of L forms are distinguished. Unstable L form, spiroplasts are capable of dividing, but can revert to an original morphology, and a stable L form that are unable to revert to the original bacteria. Like what we have seen as a protoplast or spiroplast can get back to structures, but L forms are something which are irreversibly they lose the cell wall because of the situation of the mutation, and they are called as L form because they were introduced after the Lister Institute in London. Then you have something called as the mesosomes. I think you have seen, I don't know if you remember the figure here. I'll just get back to you for the figure. Can you see this figure? Can you see the mesosomes here? I was mentioning you that there is mesosome because there are no cell organelles in a prokaryotic. You have something as a replacement for that. That replacement is called as mesosome, which is a replacement for a mitochondria. Okay. Plasmid or episomes are extra nuclear genetic material important for drug resistance. They are called as plasmids or episomes. Then you have a bacterial capsule. It is the outermost cell-defined layer in the capsulated organism, while if ill-defined, it is called as the glycocalyx. Okay, and it is in vivo structure mostly formed from polysaccharides, except in bacterial anthracis, it is formed from polypeptides. Please mention these points in your notes. All the bacterial cell walls, all the bacterial capsules are made up of polysaccharide, except bacterial anthracis, which is formed of polypeptide. That means it's a protein capsule. When it is well defined, thorough, and uniform, it is a capsulated organism. When it is ill defined, it is called as glycocalyx. So when it is thorough, it is capsule. When it is disturbed, it is slime layer. The slime layer is nothing but also called as glycocalyx. Again, important capsulated organisms is Streptococcus pneumoniae, bacterial anthracis, and Klebsiella. Then you have got flagella. You know that based upon the location of the flagella and the parts of flagella, you have got monotrichous, lophotrichous, periotrichous, amphitrichous flagella. Structure of a flagella, you know that flagella is made up of proteins called as flagellin, and the flagella is derived from an outer membrane, inner membrane. You get most of the bacteria which are mobile are gram negative, and that's how the flagella arises from the inner membrane. Then you have the Fimbriae. Then you have got the pili. Pili is, I can say, Fimbriae will help you for, Fimbriae basically helps for sometimes for locomotion, but pili is basically used, flagella also does locomotion, but pili is not for locomotion. Pili is for transfer of the material, for the cell division, for the asexual reproduction. You use pili. Okay, pili is too small actually. If you see the figure, first size flagella, then Fimbriae, then pili. Then you know the spores. You know that the spore contains a core part. You have got a thick outer membrane in the membrane pore, and then you have got shape and position of the bacterial spore. You have a central position. You have a suboval terminal. Can you please understand the location of the spore in a bacteria could be bulging or non-bulging. In that, it can be terminal, subterminal, spherical, or central. Spore has got an outer membrane, an inner membrane, and inside core. Then you have got a bacterial growth curve. This is also one of the important part of the multiplication of bacteria. You have got a lag phase, you have got a log phase, you have got a stationary phase, and you have a decline phase. The bacterial growth curve, first of all, whenever the bacteria, you understand, right? When you put the bacteria, first the bacteria will lag. That means first they will try to understand that what is my situation in the culture. Then they will start dividing. Anyone, suppose you have joined a school or a college, initial 15 days, you are new to it. You are trying to understand what is happening. Then you make friends. Similarly, the bacteria will divide. It becomes logarithmically high, exponentially very high. Then, after one limit, you stop. That is stationary phase. And after that, the bacteria, because of the competition, few of the bacteria, quarter of the bacteria, are not able to get the food to survive. Then they start declining. There's a death phase. So you have lag, you have log, you have stationary, you have decline. Again, important question. What are the four types of gene transfer in a bacteria? You have got transformation, transduction, lysogenic conversion, and conjugation. You have got transformation, transduction, lysogenic conversion, and conjugation. We will not go in detail with them, but then just for your information, transformation is based upon the Griffith concept. I don't know if you're giving maybe a AIMS or these exams. They may ask you that the Griffith concept or based on Griffith concept, which genetic transfer happens in a bacteria? Answer is transformation. And that concept is basically there is a free transfer of free or a naked DNA. There is a free transfer of naked DNA. Transfer of free DNA transformation. Then you have got transduction. Transduction is bacteria to bacteria transfer using a phage. I don't know, some people pronounce as phage. Phage is basically bacteriophage. And that is a very important step, is transduction. You all should be thorough with that. And that bacteriophage is basically a virus. A virus which helps the bacteria to transfer to other bacteria is called as bacteriophage. that virus is called as phage, whereas the process is called as transduction. Then you have lysogenic conversion. You have lytic or non-lytic phase, where you have got C. diphtheria. So most important among all the four, whatever four I have mentioned you here, conjugation, I think all of you remember, I think you have read in your zoology, conjugation means real, like the two bacteria have to come near to them. One of the bacteria has to make the sex pili, and then they have to contact each other and they transfer the material from here to there. That is conjugation. That is as simple as that. Among the four, the question can be Griffith concept based genetic transfer is, answer is 1, transformation. Bacteriophage is involved in which genetic transfer? Transduction. Then, as I said you, there is a fertility for sex pili. Okay, you have got a bacterial conjugation. I said you how they will be conjugation. The one having the plasmid is called as plus F plus, not having F minus. F plus gives an F minus. Both becomes F plus to F plus. The pilus will draw. They has to contact, and the plasmid DNA transfers to the other recipient. So that is what it is. So you can. There's one more question, like the cell, the bacterial cell, which has got a plasmid is called or uh, is recognized as which cell? It is F plus. It's the positive cell. Then there is something called as a R factor. An R factor is an RTF. Lysogenic concept is not important. That is why I have left it like that. So you can only concentrate on transduction. R factor. R factor, you have got RTF and small r determinant for drug resistance. There is a transferable drug resistance. So you have to understand when if someone has got a plasmid, it is called as F factor. Someone has got a drug resistance factor, it is a capital R factor. R factor means it's a drug resistance factor. Either it has to be R positive or R negative. Then you have got a mutational drug resistance, which basically happens in M. tuberculosis. So mycobacteria gets the drug resistance through mutation. See, drug resistance can be of two types. Either the cell can become mutation and get a resistance to the penicillin, or the body can get to the bacteria, or it can be a transferable drug resistance, which happens because of R factor. Next is the biological characteristics of the infectious agents. What is infectivity? Is the ability to infect a host. Pathogenicity is the ability to cause the disease in the host. Ability to cause the severity of the disease is virulence. Like if I say the bacteria is highly virulent, like the SARS-CoV-2 virus is very virulent or highly virulent in US than India. Violence means that the capability of causing the severity of the disease. Immunogenicity is the ability to induce immune response in a host. Immunogenicity is something which we want the vaccine to do, because the vaccine has to induce the immune response in the host. Then you have got infectious disease, some terms like what is the infectious dose, what is the latent period, what is an incubation period. You are aware of what is incubation period, what is an infectious period, what is the reproductive rate, virulence, pathogenicity, immunogenicity is important. And I think now, because of this present situation, you are very much aware of what is an epidemic, what is an pandemic, what is an endemic, what is an outbreak. What we are presently suffering is from an outbreak, which was supposed to be an outbreak, but now it is a pandemic. So first they say corona outbreak, and now it is corona pandemic because it is affected across the countries globally, right? So that is something important. Incubation period is waiting for the symptoms to arise. Infectious period is the interval during which the host can transmit the infection. Reproductive period is the ability of an agent to, that is what is called as an RO. What is bacteremia? It's a bacteria circulating in the bloodstream. You know what is bacteremia, you know what is septicemia, then you know what is colonization. You can read the definitions. You have septicemia, you have viremia, toxemia. The modes of transmission, direct or indirect. You all know again. Present situation, it's direct. Droplet, droplet is because of your sneezing and coughing and aerosols, or skin to skin contact. Then you have got indirect. It's because of the host has used those things. Someone is going and using the same thing. That is indirect. Pratyusha, you can share the question with me later. If there is a specific question regarding lysogenic concept, you can share it with me later. Okay. Because when we take the class, we prefer that 99% of the class focuses on majority of the subject. 1%, there's always a chance that we leave one or two things because there are millions of questions asked in microbiology. And if one question is asked from lysogenic concept, definitely I agree that you have right to know that. But then it's better that we discuss it later, not during the class, because I don't know. Like first, the basics have to be very clear. Everyone is maybe into a flow of first understanding the basic microbiology, right? And out of septicemia and toxemia, which could be more harmful? Septicemia is harmful. Septicemia, see, toxemia is basically poisoning. So both of them are harmful in their own ways. But which is more harmful? Is it a question or is it your personal doubt? Because if it is a question, then difficult to answer. If it's a personal doubt, then it is irrelevant because both of them have their own value because the causes are different. So septicemia happens because of gram-negative bacteria. Toxemia can happen because of any toxin. Okay, fine. If it's out of curiosity, then I think you better focus on septicemia. I don't know if you really know what is septicemia. Is you understand or you want me to explain what is septicemia? Pyemia is pus. It is the pus producing bacteria. It's a misnomer actually. Pus producing bacteria is Pyemia. Okay. You have got direct, indirect. We have got intermediate host. Then you know what is the prevalence, incidence. Mortality, morbidity, fatality, all these are the terminologies. I think you will be more thorough in these in community dentistry, PhD, right? Inapparent infection is no clinical symptoms generated. Now what you are seeing, subclinical infection. Carrier is usually no clinical symptom, but host can transmit corona. Exotoxins are the proteins which are produced inside the bacteria as a part of growth and metabolism. The exotoxins are secreted or released into the surrounding by a medium through lysis. Exotoxins are released by gram positive. Endotoxins are the lipid portions of the LPS that are part of the outer membrane of the cell wall of a gram negative bacteria. Lipid A. The endotoxins are liberated when the bacteria dies and the cell wall breaks apart. This is what is called as septicemia. Gram negative bacteria, as I said, have got an outer membrane which is made up of lipopolysaccharide. That outer membrane breaks, and this is how it happens. Exotoxin, more of gram positive. I don't say only gram positive. And toxins, more of gram negative plus listeria. Then the protein location, then the genes, then the toxicities. People say that the toxicity of an exotoxin is high compared to gram negative. Sorry, the low toxicity. The protein, highly antigenic, host forms antibodies. They are not antigenic. They're not antigenic basically. They go into blood circulation. Vaccines are available because you have got toxoid. There's no vaccine for gram negative, especially. You can think of cholera, tetanus, and everything has toxins. Sepsis, meningococcus, pneumococcus are all gram negative. I think this is clear to all of you. Microbial culture techniques. Then I think you all should be, or maybe would have done in your second year regarding the various types of cultures and why do you do the cultures. The microbial culture is the method of multiplying the microbial organisms by letting them reproduce in a predetermined culture media. A term given to combination of all the ingredients that will support the growth and cultivation of organisms. I have mentioned you before regarding Louis Pasteur. What is his contribution? He used simple broths which are made up of urine or meat extract to grow the organism. Okay. Why do we have to culture? Because we need to isolate the bacteria. We need to look into their properties, their biochemical properties, their growth characteristics, their method of isolation, their sensitivity to antibiotics. So we need to know. And maybe the reproductive rate, the culture, the bacterial growth curve. So why do we have to culture is these are the reasons why we have to culture. Now, the methods of culturing the organisms, you have got inoculation, isolation, incubation, inspection, and last, identification. Equipment used specifically is a petri dish, a Bunsen burner, and an inoculating loop. We have got various ways of aerobic and anaerobic cultures. Streak, lawn, stroke, stab. These are all aerobic culture media. Got an anaerobic culture methods, and I think everyone should be aware of what is McIntosh Field's jar, where we use gas pack. You have an anaerobic jar, and I think you all should know that the anaerobic culture example is Robertson cooked meat broth and thioglycolate broth. These are very, very important two examples of an anaerobic culture media. This is a streak culture most commonly used for isolation of the bacteria. You can use a simple agar plate and you can make streaks and grow the bacteria. The bacteria grows. You can see the growth pattern. You can take a maybe you can this is the way you make streaks. Yeah, this is not important now. Advantage of streak culture is for isolation of the bacterial culture. The disadvantage is there's a high probability of contamination prior to isolation. Streak plate method can be used for qualitative and non-quantitative studies because it cannot be used for enumeration of appropriate number of bacteria. It is just a very vague method of culturing it, but a simple method. Lawn culture, also called as carpet method, provides uniform surface growth of the bacterium and used for antibiotic sensitivity also. This is for lawn culture. Then this is a stroke culture. You can use it for agar slope, employed for providing growth of bacteria for slight agglutination and other diagnostic tests. Tap culture. You give a stab onto the broth and then you grow. pour plate, liquid cultures, and the most important, anaerobic culture method, production of vacuum, anaerobic culture method. Initially, there was a candle jar because that used to create carbon dioxide gas and displace the oxygen from the other gases and make it an anaerobic media. But nowadays, you have got other chemicals which are used for causing anaerobiasis. One is called as the pyrogallic acid. It was Buchner who was the first one to use alkaline pyrogalol for anaerobiosis, which absorbs the oxygen. Or you can use a combination of chromium and sulfuric acid. Mixture of powdered chromium and sulfuric acid can be used for anaerobiosis with jars or with the yellow phosphorus. It forms chromous sulfate and absorbs oxygen. This is the McIntosh-Fields jar. This is a very important question. Anaerobic medias are important. Why? Because of in dentistry also, most of the periobacteria are all anaerobic. That is why even your P. gingivalis and other bacteria, you can also grow them in all this type of jars. So these jars, you put a catalyst or you put a gas pack, it displaces the oxygen from that, makes it anaerobic in nature, and the bacteria grows. And I think you all have already, maybe you remember that I just mentioned you in the previous slides, that anaerobic jar is used just to create the anaerobic environment. But the culture method or the culture media is either Robertson cooked meat broth or pyroglycolic broth. Thioglycolic broth. Gas pack. It is commercially available. It contains chemicals which generate hydrogen and carbon dioxide on addition of water. It is a very, very important question. It's a commercially available disposable envelope, disposable hydrogen and carbon dioxide generator envelope, H2 and CO2. It is very, very important. The composition, the name, and the way it is marketed in a disposable envelope. You have an anaerobic chamber also, maybe on a larger scale if you want to culture something. Other methods, you have got reducing agents, you have got broth. These are the various culture medias. I told you a basic culture media can be an agar. It contains electrolytes, some peptones. If you add peptone to water, it's called peptone water. Some meat extracts, blood agar, chocolate agar, serum agar. Whatever you add to agar, it becomes something enriched with that. So you have got a solid state agar. When any culture media is solid, it is agar. When liquid, it is called broth or semi-solid. It could be basal, simple like agar, complex or synthetic. As I said you, in special you have got enriched, enrichment, indicator, selective, transport and sugar. The first media which was introduced by Robert Koch was cooked potato. Later, gelatin was introduced to solidify the liquid media. Agar was first introduced as a solidifying agent because agar is, you know what agar is, right? Liquid media is broth, you have got water, you have got peptone water. Semi-solid media, then you have got a simple, which is called as a nutrient broth. It just contains peptone water and meat extract. When glucose is added, it is called as a glucose broth or a glucose agar. When agar is added, it is called a nutrient agar. Complex, you add peptones to it. Synthetic, you add some chemicals to it. This is nutrient agar. This is nutrient broth. This is enriched media. When the basal media is added, with some nutrients like blood agar, serum, or anything, then you can do it like that. You have got blood agar, you have got chocolate agar. It may be used for growing a number of bacteria. Chocolate agar, you add chocolate, that means heated blood agar for isolation. And you can use for Neisseria or H. influenza species. Leuphus serum slope, very, very important because it is the only media for isolating Corynebacterium diphtheria. Please mention in your notes. Leuphus serum slope is for C. diphtheria. Enrichment media, like some substances that are incorporated in the liquid media that have a stimulating effect on the bacteria grown. Enrichment is the tetrathionate broth, selenite F broth. Then you have got a selective media, like they contain substances that inhibit all but few types of bacteria, facilitate isolation of the particular species, like DCA. Okay. See, if it is a solid, it is called agar. If it is a liquid media, it is called as a broth. When you have a simple nutrient agar or a nutrient broth, nutrient agar and broth, difference can you see? Solid is a plate form, in a test tube, it is a broth, liquid. When you add something to the basic media, you call it enriched. It can be blood, it can be chocolate, it can be serum. When you add something into it, especially liquid media, which will stimulate specific bacteria, then it is called enrichment media. Example, you should know: tetrathionate broth, selenite F broth, or alkaline peptone water. I am using the word broth, that means it's liquid. Containing substances that will inhibit all but a few types of bacteria and facilitate isolation of them is called as a selective media. Example, you have got deoxycholate citrate agar. Then you have got a differential media. When a media contains substances which helps to distinguish different bacteria, that is called very, very important, McConkey's agar. No, 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 no. All the broths cannot be enrichment media. Why? You can have a nutrient broth also. You have to understand that. See, you can have a simple nutrient broth. You even peptone water is a broth. Serum slope is a broth. If you are adding something into it which will enrich only one family of bacteria to grow, then it is enrichment media. Okay, clear, Lakshmi? Is it okay? That is the difference between that. Broth means liquid. Broth can be broth can be enrichment. It could be selective. It could be differential. It could be indicator. Whatever it is. This is again very important. Media containing an indicator that changes the color when the bacteria grow. Example: Wilson-Blair media. The indicator. And again very important, which is the only culture media which is used for transport. It's called as Stewart's transport media. I have told you about anaerobic media. Please remember these two words: Robertson cooked meat broth and thioglycolate broth. And maybe when I do mycology, I will talk to you about SDA media, Separos dextrose agar. That's the only media which is used to grow fungi. SDA media. Significance of uncultivable bacteria. There are some bacteria which you cannot cultivate. So these bacteria have got around their own system. These are the two antibiotic sensitivity tests: diffusion test and dilution test. You should know the names of the tests they use for antibiotic sensitivity. In dilution, again, you have got two types: tube dilution and agar dilution. So, are you all fine for me to continue, or you want a break for 5 minutes? Do you want a break for 5 minutes, or shall I continue? Okay, okay. Then I'll give a break for 5 minutes. After 5 minutes, we'll start. Okay, no problem. Fine. Can we start? Can I start? Okay. Let us start with disinfection and sterilization. You all are aware of the sterilization and disinfection, the basic differences between both. And sterilization is a process where article surface is freed of all the organisms, living or even sporotic form. Disinfection is destruction or removal of all the organisms capable of giving rise to infection, but not the spores. And nowadays it has become a trend to use word sanitization. Nothing but like disinfection, but particularly used with reference to food processing and catering in microbiology. But nowadays we are using for even for clinics sanitization. Antisepsis is used to indicate the prevention of infection, usually by inhibiting growth of bacteria. Bactericidal agents, bacteriostatic agents, all these are used to prevent the infection by inhibiting the growth of the bacteria or by killing the bacteria. There is one more classification, Spalding system. I think this is uh, there in your conservative dentistry textbook. Critical, semi-critical, and non-critical. Yeah, critical. You have semi-critical and you have non-critical. Heat is the most commonest, cheapest way of sterilization. Fast, inexpensive, definitely. Factors determine whether the heat is dry or moist, presence of organic matter, number of organisms present, physical control with heat. A dry heat, you have got direct flame, Bunsen burner. Incineration is something where you burn them and then you just cover it with mud or somewhere, all the shafts and all. Then you have a hot air oven, radiating dry heat for 160 degrees and 2 hours. Useful for sterilizing dry powders and other things. Please remember 160 degrees for 2 hours. Advantage, non-corrosive method. Hot air oven. Moist heat, you have got below 100 degrees, which is called as pasteurization. At 100 degrees boiling, remember these values. Boiling water. Tyndallization. Tyndallization is actually popular because it is also called as intermittent sterilization, or you can say as a fractional sterilization. There is a free flowing steam at 100 degrees for 30 minutes on 3 successive days. Means the steam is given in fractions. That's called tyndallization. Autoclave, you know the pressure cooker mechanism. You know very well 121 degrees for 15 lbs pressure for 15 minutes. You increase the temperature, you decrease the time. I don't know if you are aware that there are two types of autoclaves. I think you should be aware. Autoclave class B and class N. Okay. I don't know if all of you are already aware of these things. Class B and class N. These are the two types of autoclaves. Nowadays, I think everyone should know that. Class B and class N. Class N autoclave is something which you come across in your regular dental clinics or your colleges. The one like a pressure cooker, a simple autoclave. The only main the difference between class B and class N is class B is more of digitalized. You can do a lot of other, you can incorporate dry cycle into it. Penetration is very high, and after you sterilize, the instrument stays for at least a week time. Class N instruments come out from an autoclave in a wet form, do not have deep penetration into the instruments. You need to keep them open. So there are a lot of differences between class B and class. Class N is very cheap actually. Class B is damn expensive. Yeah, filtration, not important. You have UV light, you have microwave, this is all not important. We don't do it actually in industry. You have disinfectants, you have phenol coefficient. It is a measure of the effectiveness of an antiseptic or a disinfectant as compared to phenols. If you want a very high level disinfection, then you have got ethylene oxide gas. If you have an intermediate level disinfection, then you have got bactericidal agents like formaldehyde. Or you have a low level disinfection, They have narrow antimicrobial activity. Mechanisms of antimicrobial action. You have something called as the surface active agents. These are the substances which alter the interfaces. You have got anionic, cationic, non-ionic, amphoteric. You have got a cationic detergents. Anionic detergents. Soaps. Phenol. Phenol is very important. It's used for phenol coefficient test. Azoles. Bisphenols. Alcohol. You've got acids and alkalis. Heavy metals. I think maybe you all have heard about silver nitrate antiseptic disinfection, 1%. Oxidizing agents like halogens, chlorine, sodium hypochlorite, iodine, iodophores, hydrogen peroxide, you have got various dyes, alkylating agents like formaldehyde, ethylene oxide, and glutaraldehyde. These are one of the best disinfectants available. Formaldehyde is a gaseous form formalin. Ethylene oxide is used for cold sterilization. I think you have, maybe if you read what is mentioned on uh, the plastic cover in which your LA needles are there, or maybe some syringes are there, what do you see? Cold sterilized. If you notice that, or you can see ethylene oxide sterilization done. So those things are done with ethylene oxide. Cold, that's called cold sterilization. Glutaraldehyde, I know the very popular brand of glutaraldehyde is Sidex. It's 2 to 3.58 to 3.4% effective. These agents interfere with membrane functions. And you have instrument processing. Holding, that's pre-soaking. Pre-cleaning, corrosion control, drying, packing, sterilization, sterilization monitoring, and handling the processed instruments. How do you monitor sterilization? How do you monitor sterilization? This is how, okay, we'll go into it. But then you have got holding. Means to prevent the drying, you can use an instrument, a bucket or something, detergent and all, just pre-soak. Pre-cleaning, ultrasonic cleansers are there. Corrosion control. Packing prevents contamination, so you need to take care of the packing also. Then you have got cassettes in which you can prepare. Then you have sterilization. In which you have got 4 cycles. You have a heating up cycle. Then you have the sterilizing cycle. Then you have the depressurization. And then you have the drying. You heat because you convert water to steam. That steam will penetrate inside. That is what sterilization. Then you decrease the pressure, depressurization cycle. And then comes the drying. This is a very important question. Arrange the following steps of sterilization: steam sterilization or moist heat sterilization in order. So first is drying. No, sorry. First is heating up, then sterilizing, then depressure, and drying. This is your class N sterilizer. You have something called as chemiclave, where with the steam you use a solution like 0.23% formaldehyde and 72.38% ethanol plus acetone. Same thing happens, but instead of pure steam, there will be a chemical steam. It's called chemiclave. Again, important question. Temperature remains same, but chemiclave. Then you have dry heat. Gas, I told you, ethylene dioxide. Very, very low temperature. Liquid chemical sterilization, very common is butadiene. So, what is popular is butadiene, steam sterilization, ethylene dioxide for few of the plastics, or pre-packed surgical instruments, your gloves, surgical gloves. Then you have glass bead sterilization. This is something very important. This is what is called as sterilization monitoring. You have got biologic monitoring, chemical monitoring, and physical monitoring. Very, very important. Please write this names of the bacteria. You have biologic monitoring. You have got chemical monitoring. Maybe I don't know if you have noticed the autoclave tapes. And then you have got physical monitoring. When you're using the autoclave or steam sterilizer, you use the spores of bacterial steatothermophilus. Otherwise, for dry heat or ethylene oxide gas, you can use Bacterius subtilis. Basic immunology. This is something which is very volatile, very dry, but one of the most important topics in NEET exam. Immunology. Anyhow, we will not go into the various depth of the cycles of the immunity cycles, but then yes, the basic immunology, yes, you should be thorough. We will talk about all these things. I think you all are aware of what is innate immunity and what is adaptive immunity. Innate is something which is inborn. Adaptive can be natural or artificial. Okay? Natural or artificial. You may get the immunity naturally from your mother. You're getting it naturally from mother to the fetus, which is passive. When you're getting the immunity by getting the infection, measles, mumps, chickenpox, you are getting the infection, you are suffering with the infection, and then you got the antibodies. So that the next time the virus doesn't attack you. That is natural active. Then you have got artificial immunity, which can be again passive, like ready-made antibodies, or active, which is immunization. These are your lymphoid organs. From adenoids, tonsils, thymus to various primary organs and secondary organs. The primary organ definitely is bone marrow. Secondary are a lot of you've got Peyer's patches in intestines, you've got lymph nodes, inguinal, clavicular, you've got so many. Then your bone marrow, the site of hematopoiesis, thymus, lymph nodes, spleen, Peyer's patches. You have got neutrophils. Neutrophils, as you can see, these are the granulocytes. Multi-lobed nucleus has got primary granules called as eosinophilic granules. Secondary granules are there. Primary granules are there. Secondary granules are there. This is how your phagocytosis happen. First, there will be opsonization, chemotaxis, Phagocytosis. Phagosome fused with lysosome forms phagolysosome. Neutrophils have got two types of granules. You have got specific granules contains lactoferrin lysozyme. Azophilic granules, which are mainly for the intraphagolysosome secretion. You have got eosinophils, which are basically they are responsible to release histaminase and aryl sulfatase. Inactivates the mast cell products histamine. Antiparasitic and antihelminthic activity mediated by IgE. Eosinophils will increase because of these things. Eosinophils. You've got basophils, bilo. You've got two types of mast cells, mucosal mast cell and connective tissue mast cell. They possess a receptor for complement components like C3A and C5A, as well as receptors for Fc portion of an antibody molecule, IgG and IgE. So whenever the anaphylaxis has to happen, the C3A, C5A will fuse with the receptors and the mast cell degranulation will happen and releases vasoactive amines like histamine and serotonin. So as I said, histamine or a degranulation of mast cell, histamine is released, and what does it do? Basically causes vasodilatation, which is the primary source for vasodilatation in the vascular event of inflammation. These are the mononuclear phagocytes. Starts from stem cells, monoblast, monocyte, and monocyte in the tissue becomes a macrophage. And you know that macrophages can become microglial cells in CNS, Kupffer cells in liver, alveolar macrophages in lung, osteoclast in bone. It can do anything, and it's got various names based upon the location. A monocyte and a macrophage. Macrophage is basically useful for pseudopodia, for what do I say, phagocytosis. These are the cells of chronic inflammation. Neutrophils are cells of acute inflammation. Macrophages are APCs, antigen presenting cells. Phagocytosis. They clear up the debris, the dead tissues. They are delayed hypersensitivity. Then you have got dendritic cells. These are also APCs. Then you have got B lymphocytes and T lymphocytes. B lymphocytes are nothing but responsible for secreting immunoglobulins. Then you have got T lymphocytes, which mature in thymus. You have got Th1, Th2, Th3. Then you have got natural killer cells. They possess several classes of antigen receptors. You have got KIR and KAR receptors for NK cells. Then you have innate immunity and adaptive immunity. Innate immunity means your body defense, your epithelium, your saliva, complement, your NK cells, phagocytosis. All that is part of your innate immunity. Adaptive immunity, where your B cells and T cells will play an important role. Adaptive. Got innate immunity and acquired. So you have got physical barriers, skin mucosa, complement, phagocytes, or an inmate, but it doesn't have. Hello, can you hear me? Now, am I audible? Okay. Innate immunity is based on your species, your race, your age, your nutrition, and there are a lot of protective barriers. You have got anatomic barriers, physiological barriers, phagocytic barriers, and inflammatory barriers. Anatomic barriers, epithelium, most important is epithelium. Physiologic barriers like temperature of your body, the low pH. Chemical mediators, you have got complements, you have got cytokines, you have got TLRs. Complement system, I think you all know that it is 30 membrane associated cell receptor proteins. The last slide. Yeah, you have the anatomic barriers. Then you have the physiologic barriers. In that you have got the temperature, the low pH, and the chemical mediators. The chemical mediators, like you have got complements, cytokines, TLRs. These are the chemical mediators. Complement system. You have a vasoactive substance, kinin like C2A induces pain and increases the vascular permeability. And I have said you what is anaphylatoxin, C3A, C5A. Chemotaxin, C5A attracts phagocytes. Opsinin, You have got cytokines. These are the polypeptides which are present, and you know what are cytokines. You know that there are cytokines which are based upon innate immunity, based upon adaptive immunity. In detail, it is not necessary. Then you should know what is TLRs. These are the pattern recognition receptors which are present, and they're recognized by PAM, pathogen associated molecular patterns from the bacteria. They're first identified in Drosophila. You have a physiologic barrier. Then you have a phagocytic barrier, where there will be phagocytosis by macrophages, monocytes, engulfment, phagolysosome, killing. Vasoactive amines, neuropeptides. Innate to adaptive immunity. Spores antigen, dendritic cells, adaptive immunity. Adaptive immunity. You have got humoral immunity, which is by B cells, and cell mediated by T cells. B cells is basically for extracellular microbes. T cell is for the intracellular microbes. You know what is active and what is passive, right? Then you have the cardinal features of immunity. You have specificity, you have diversity, you have memory, you have specialization. You know what are antigens, what are haptens. They're antigenic but not capable of producing the immune response. Immunogenicity. Epitopes are immunologically active regions of immunogen that bind to antigen-specific membrane receptors. You have MHCs. MSCs are nothing but the presentation way. You have got MSC class 1, MSC class 2. MSC class 2 are present peptides to CD4, MSC class 1 to CD8. Antigen presentation cells. Don't get confused. There's a lot, but then this is not important for you. Antibodies, 2 heavy chains, 2 light chains. Immunoglobulins secreted by B lymphocytes. Antibody mediated immunity or humoral immunity. 2 heavy chains, 2 light chains. Light chains can be kappa or lambda. Heavy chains can be 5 types. Based on that, we have got names. You have got a variable zone and a constant region. The C region is called as a carboxy terminus. The V region is called as an amino terminus. IgM pentamer, J chain is there, extra. IgG is a single monomer. IgA is a dimer. IgE is again the monomer. IgD is again I can say it's another part of IgG, but not a specifically strong antibody. Then you have got hypersensitivity reactions. Immediate is, and this is very important, Coombe and Gell, the name of the person. Coombe and Gell have classified the hypersensitivity reaction into 4 types. The immediate is 1, 2, 3, and the delayed is type 4, which is by T cells. Type 1 is nothing but anaphylaxis. There will be an antigen, there will be an antibody, there will be anaphylaxis, there will be mast cell degranulation. You have an IgE Fc receptor and mast cells will degranulate. This is what they will release. The primary mediators of anaphylaxis, histamine, serotonin, and eosinophilic chemofactor of anaphylaxis. The secondary mediators is SRSA. SRS is slow reacting substance of anaphylaxis. SRS full form. PAF is platelet activating factor. PGs is prostaglandins, and TXA is from vaccine A2. What is the shock organ is lung. Types is in vitro anaphylaxis is called as Clotesdale phenomena. An important question. And cutaneous is wheal and flare. Atopy is hay fever or asthma. Pollens, house dust, atopins inside the atopy is like a small, I should say, a younger brother of anaphylaxis, like asthma, which happens with pollens. Atopins induce IgE antibodies. Type 2 hypersensitivity is called as a cytotoxic reaction. Type 3 hypersensitivity is called as antigen antibody complex reactions. Type 4 you have hypersensitivity reaction. Sometimes people call as type 5, which is stimulatory type of reaction. Schwarzman reaction is related to Salmonella typhi. Then autoimmunity. Then this is your congenital immunodeficiencies because of phagocytes, because of lymphocytes, and acquired. Did you really understand what is the basics of immunity? Did you all understand that? Did you understand? See this Church-Gale anaphylaxis reaction is basically it's basically to promote anaphylaxis or completely blocking it. So if you block the histamine receptors and serotonin receptors, you will not see the anaphylactic clinical reactions. That is the most important thing about it. What I want you to understand is what is the role of B cell, See, Acha, you want to know about herd? Herd immunity is an indirect immunity which the population will be able to gain from the infectious disease when it is contagious. When the sufficient population is able to get it or immune to the infection, then it is called as herd immunity. And actually speaking, herd immunity will happen through vaccination. Maybe nowadays people are talking about it, but then herd immunity means when maximum people in a population are affected actively or through vaccination, when the majority of the population is having immunity towards the contagious disease, then it is herd immunity. Nazia, I don't mind to repeat if you really want to understand anything in immunity. Can you please let me know what exactly is the problem? Nazia, you want me to explain about T cells, B cells, active, passive? What exactly? Can you please let me know? You want to know about cytokines, right? Cytokines are nothing but proteins which are released from the cells and which act on the other cells. Cytokines are something which are classified based upon the function and based upon the family. Cytokines can be interleukins, cytokines can be TNFs, cytokines can be interferon, cytokines can be a colony stimulating factor based upon what cytokines are doing. If cytokines are responsible for immunity, you have IL-1. If they're IL-1 and TNF. If cytokines are responsible for colony stimulating factor for your blood cells, interleukin-3. If cytokines are responsible for chemokine activity, interleukin-8. So cytokines are classified based upon their function. Cytokines are proteins and it includes interleukins, TNFs, interferons, colony stimulating factors. Is it clear or you want me to repeat again? Okay. B cells. B cells are lymphocytes. B cells are WBCs, which are A granulocytes. They don't have granules and they are lymphocytes. You have two types of lymphocytes. You have B cells and T cells. B cells are nothing but the cells which are responsible to secrete antibodies. B cells secrete antibodies. So the immunity the B cells provide are antibody mediated immunity, AMI, which is also termed as humoral immunity. When the T cells fail to do their function, then B cells will act because they release antibodies. So B cells will form plasma cells and that will release antibodies. That is the function of B cells. Okay. Anything else, Nazia? Okay. Anything else? Did you all really understand what is APC? Well, I want you to understand what is APC. What does an antigen presenting cell do? The macrophages, the dendritic cells, they present the antigen to T cell. And in T cell, you have got T helper, you have got TC, you have got TS. TC, TS, TH are the three types of T cells. And what T cells do is cell mediated immunity. They directly kill the bacteria or the organism by their immune responses. If they fail, it goes to B cell. Anything else, or shall we conclude for today? Because the next class, maybe tomorrow, I will be talking about virology, mycology, the specific function of T cytotoxic cell is the T cytotoxic cell will kill cytotoxic. That means it kills the opposite cell. T suppressor and T helper are two types of cells, what you called as your CD4 and CD8. So these cells will suppress, or T helper is helper cells. T helper means it is basically used for using the cytokines and helping the immune cells to fight with the foreign body. You have got in that also you have got Th1 and Th2. T cytotoxic cells are basically used for killing the cells directly. Okay. T helper cells are your CD8 T cells, like CD4 T cells. These are the cells which are basically having the co-receptors, and they will present it to the B cell. T helper means it helps to present it to the B cell. B cell is the last cell which is going to fight. If it is done with T, it is done. If it is not done with T, it goes to B. First, who will fight? Body. Innate, inflammation, complement, then goes to APC. From APC, T cells are again there. That presents to B. B will fight through antibody. This is the story. Is it okay? See, T1 cells will stimulate the immune response. T1 cells will stimulate the immune response. Th2 will stimulate the humoral response. You able to understand? Th cell, Th1 cell, when the T cell is there, that will stimulate the immune response. Which immune response? Itself. Th will give it to the other T cells. It's helping to T. T cells are helping the other T cells to do the function is Th1. Th2 is the loser. It is giving when the Th1 fails, Th2 takes it and gives it to B. That is why the Th2 is specifically antigen presenting to B. That is a function of Th1 and Th2, sir. Is it okay, sir? Okay, sir. Thank you. Yeah. See, prozone phenomena. Okay, very good. Prozone phenomena is basically used to test the antibody's action. So whenever there will be what do I say, a high, maybe proson phenomena and all, you can see in Vidal test. So when there is a very high antibody titer, when there is a very high antibody titration present, it will affect the antigen antibody reaction. You understand? The antigen has to fuse with the antibodies. When the antibodies are more when compared to antigen, the antigen antibody complexes may not form. It becomes impaired. That is what is proson phenomena. That will appear in front of you in the test tube when you do a test. That is what is proson phenomena. You do it for Vidal, you do it for syphilis, and you can see maybe what do I say, a cotton wool, or you see a lattice-like network. That is what is proson phenomena. So maybe for you, proson phenomena is associated with which aspect of immunity? Answer is impaired antigen antibody responses or an antibody defect, especially when the antibody titer is more. Okay, anything else? Is it okay? Shall I end the meeting? Yes, yes. It means that antigens are less than antibodies, which is a false negative. It is not, it doesn't happen. But in terms of false negative results, sometimes that happens, and that is why the antibodies get impaired. The reaction get impaired. More antibodies, less antigen will lead to proson phenomenon. Okay. So I think we'll be having the next session tomorrow, but in the afternoon or the evening time, I will just let you know the time. Okay, then I shall end the meeting then.